Hello, everyone. It is my great pleasure to introduce our oncology seminar speaker today. Dr. Jinghui Zhang is the inaugural chair of the Department of Computational Biology and endowed chair of bioinformatics at St. Jude Children's Research Hospital. During her early career at NCBI, she helped develop the most popular DNA search algorithm, BLAST. Later at St. Jude, she developed innovative computational tools for integrative analysis and visualization of somatic mutations and germline variants in cancer. She mapped the germline cancer susceptibility in pediatric cancer patients and survivors, leading to a new comprehensive new recommendation to screen for germline susceptibility mutations and received the 2019 AACR Team Science Award. She pioneered in the detection of structural variations and recurrent mutations in pediatric tumors and carried out the first comprehensive pan-pediatric cancer analysis, unveiling major differences between pediatric and adult cancer genomes. She developed an integrative clinical sequence platform to incorporate whole genome sequencing with pediatric oncology testing, which led to the development of new therapeutics in pediatric cancers. She also developed the St. Jude Cloud, an ecosystem for accessing, analyzing, and visualizing 1.2 petabytes of omic data from over 10,000 pediatric cancer patients, which received the Microsoft Healthcare Innovation Award. So before I turn the podium to Dr. Zhang, during the talk, if you have any questions, you can type it in the questions and answer section. Please don't type it in the chat, so type it in the QA. So without further ado, let's welcome Dr. Zhang.
Oh, thank you, Sherry, for inviting me to give a talk at Dana-Farber, and I'm really excited for the opportunity to, connecting with my colleagues in pediatric oncology and computational biology to share our experience in analyzing genomic variants in pediatric cancer. And so today, as the title suggests, I will talk about our mathematical modeling for relapse, precision oncology, and data sharing ecosystem. Okay, when we analyze genomic variants, usually we start the journey with a research cohort, which we perform molecular profiling. From there, we define diagnostic and prognostic markers so that we can stratify patients into different treatment protocol. So as the clinical genomic profiling become more common, the research actually can start utilizing the data generated from a clinical setting, which will start a new iteration that will enhance our knowledge of how genomic data contributing to the diagnosis and prognosis of the disease. So we started this journey in 2010 with the launch of St. Jude Washington University Pediatric Cancer Genome Project. We call it PCGP. So we profiled the major subtypes of the leukemia, solid tumor, and brain tumor, performed the paired tumor normal whole genome sequencing, exome and RNA-seq. And we also performed extensive experimental verification. And these data not only lead to major discoveries of over 20 subtypes of pediatric cancer, but also enable us to develop very accurate computational analysis tools, which we later deployed for clinical utility that allow us to moving forward from the research to clinical. So as Shirley mentioned in her introduction, so our group also collaborated with National Cancer Institute, Target, to carry out the first pan-cancer analysis of pediatric cancer. And I want to particularly mention that Target was the brainchild of a dear friend and colleague, Daniela Gelhardt, who was the NCI Office of Cancer Genomics, the director of the Cancer Genomics, and unfortunately she passed away 2 months ago, suddenly and unexpectedly. So whenever people, when at least like me, using a target data set, I always remember the legacy of Daniela, always stay with us, and she's the one really pushing forward, indicating that pediatric cancer need to be treated with the same level of attention as adult cancer. So the target data set is comprised close to 1,700 patient samples, as shown here in 6 major subtypes: B lineage AL, T lineage AL, AML, neuroblastoma, Wilms tumor, and osteosarcoma. And these samples were profiled by whole genome, RNA-seq, and exome sequencing. And I want to mention that the whole genome sequencing data was generated from the complete genomics platform. And in very early days, these data actually contain a high number of the PCR artifacts. So what we have done is to perform extensive data filtering, which start with 100 million candidate somatic variants delivered by the company to filter and perform manual curation. That goes down to about 44,000 coding variants after these steps. Using these, we're able to perform statistical analysis, which led to discovery of 142 driver genes. We have also performed the mutational pathogenicity classification that lead to the discovery of 82 additional driver variants with P or LP variant classification. So one of the striking findings we see is that of the 102 new drivers we found, the majority of them, 55%, were absent in 3 adult pan-cancer studies. That really showcased that the molecular drivers in pediatric cancer are distinct, and you cannot really treat a child like a small-sized adult. And the second part is we found that 62% of the driver variants were structural copy number variations. And this again highlights the importance of employing whole genome-based approach to profile pediatric cancer genomes, either in the research or clinical setting. So these are the major biological pathways altered in pediatric cancer in these 6 subgroups. And we have color-coded here those that were the percentage of the genes in each pathway that were unique to pediatric cancer. As we can see that transcription factor is a major category, the #1 category of genes being affected by somatic alteration in pediatric cancer. And the majority of the genes are distinct from the adult cancer. And that really leads to some of the interesting, thoughts about how we can target transcription factors, whether using degradon or other approaches will be the way of, targeting these events. So this study was led by Shar and Yu in my lab, and both of whom now have their own independent laboratories, and with the support from the NCI target team, as well as multiple lab research groups at St. Jude and Vladimir Alexandrov from UCSD, as well as Robert Huser, who's a former postdoc fellow who's now at the Tempus. And the study was supported by National Cancer Institute. Now I want to go to a little bit more detail, talk about the our study on drug-resistant mutations in relapsed ALL. And this is a collaboration with Shanghai Children's Medical Center. So for this study, we were able to profile 103 patients utilizing the trio samples collected at the diagnosis, relapse, or remission, which serve as a germline for these. We performed both whole genome and RNA-seq. These 103 patients can be categorized into 3 groups based on the time of the relapse. The very early relapse groups, relapse between 9 months, and early relapse between 9 to 36 months. These are the relapses during treatment, and the late relapse was post 36 months, late relapse after 36 months. So we compared the mutations derived from diagnosis and relapse and find that 12 genes listed here appear to have enriched mutations in relapse, as shown in the purple or red dots here. So what is really striking is that all 12 genes were involved in drug metabolism pathways in drugs that are known to be used for treating pediatric cancer, like glucocorticoids, thiopurine, and methotrexate. And p53 is a gene has a broad spectrum resistance to these multiple drugs. We also realized when comparing these three groups, the early relapse groups shown in middle here has a significantly enriched mutation content compared to very early and late relapse groups, suggesting that drug treatment may play a role in generating these resistant mutations. We then looked into the mutational signature at the diagnostic and relapse samples. And these y-axis are referring to the number of the somatic mutations identified in each tumor samples. And we also color-coded these by the cosmic signatures that we identify in these two cohorts. So I think what we have discovered is that there are two novel signatures, what we mark as blue and magenta here, A and B, that only are present at the relapsed samples, but absent in diagnosis. And in relapse samples, they are only present in the early and late relapses, but not in the very early relapses, suggesting a potential connection to therapy again. And there's resistant mutations appear to be associated with both novel A and novel B signature, including PRPS1, TP53, NT5C2, and NR3C1. Next, we decided to do an in silico experiment trying to find out which drugs may be responsible for inducing these novel signatures. So we used the ICGC Peacock cohort for performing this and the NCI's target cohort to perform this experiment. So what we found is that both novel signatures appear to be absent at the diagnostic cases in both the adult Peacock cohort and the pediatric target cohort. For novel signature A is almost only present in the Chinese cohort, where the novel signature B are common in both the target and Shanghai cohort. So what we suspect is novel signature B may be related to the use of thiopurine, which is a purine analog that was used for the maintenance treatment of ALL. So to prove that, we performed a wet lab experiment. First we did is to use the ALL cell line REH and expose it to thiopurine, and then isolate single clones and perform whole genome sequencing and looking at the mutations that present only in the treated cells, but absent in the primary diagnostic samples. So each of these bars representing a mutation detected in these single cell clones. So what we have found is somewhat disappointing in that what we see here is a lot of shared mutations among these clones, suggesting that treatment is only isolating these resistant clones that are already present in the initial ancestor of the REH. We then realize REH, even though it is an ALL cell line, is actually not a good model for this experiment, because like most of the other ALL cell lines, it was derived from a relapsed case. So the patient samples used to create the cell line is already resistant to the ALL treatment drug. So then we take a bold approach of using the normal breast cell line, MCF10A, as an experimental model, repeated the same process. We chose MCF10 because it was the cell line that was used to prove cisplatin-induced mutational signature in breast cancer. So this time, in addition to thiopurine treatment, we also treated the cell line with cisplatin so that we can have a positive control of this experiment. And we can see the result is already look very good compared with the REH, as we can see very distinct private mutations in each of the single cells. So then we performed the mutational signature analysis, and we were able to get our positive control working, cisplatin signature generated from clone 5 with very high correlation with the standard cisplatin signature published before. And what is more exciting is that we indeed was able to replicate the novel signature B identified in the patient samples with our in silico, in the cell line model with cells exposed to thiopurine with very high correlation coefficient. So this experiment indeed confirms that thiopurine is the drug that responsible for inducing the mutational signatures we observed in the relapsed AL. So this study really allow us to think differently about how relapse occur in cancer. So the prevailing two models, one is a de novo resistance, which was not observed in ALL because our patient went through remission. The second is a chemo selection of a very early relapse of a clone leading to relapse. And this is actually account for the very early relapse cases we observe in ALL. So our new model based on these data would suggest that ALL relapse went through two-step process. First is that clones have to survive the chemotherapy, and these are what we call the persistent clones. And then during treatment, they will acquire chemo-induced mutation and leading to clonal expansion at relapse. And we believe that most of the early and late relapse cases could be explained by this model. So the study was published early last year, and most of the mutational analysis was done by Sam Brady in my lab. And he also designed and performed the cell line treatment experiment, confirming the origin of these novel signature A and B, and which now become the cosmic mutational signature 86 and 87. So even with the publication of the first study, that we still have several unsolved mysteries. And the first is that we did recognize there's a fair number of the ALLs acquiring DNA mismatch repair mutations at the relapse, and some of these were biallelic. However, we did not find any of these DNA mismatch repair signatures in any of these cells. So it is a puzzle. Secondly, when we look at the p53 mutation acquired at the relapse, we recognize there's a very strong mutational hotspot R248Q, which is absent at the diagnosis and also absent at any of the other primary tumor samples sequenced, adult or pediatric sequenced up to date. So what is the cause of this? By looking at the TP53 mutational sequence context, we recognize that those mutations acquired at the diagnosis, this is their profile, and those are the ones at the relapse. So we can see that at a relapse, the R240Q signature is quite distinct from those that we see at the diagnosis. We then look at the full spectrum of the ALL mutations at diagnosis versus those that have the thiopurine signature, and that we can, and also at the diagnosis, we do see one sample that have a DNA mismatch repair deficiency, and they indeed have this cosmic signature 26 representing DNA mismatch repair deficiency here. And in those that we have MMR deficient at the relapse, this peak was clearly absent. So we hypothesized that it is possible that when cells acquire DNA mismatch repair during treatment, coupled with thiopurine treatment, may generate a unique signature. So to prove that, we resort to back to our MCF10A experiment. So in addition to exposure to the wild type, which wild type MCF10 with thiopurine, which is an experiment we did in the first study, we have also knocked out MSH2, a gene involved in DNA mismatch repair, and then exposed them to thiopurine. So we can see that in this experiment, the wild type replicated thiopurine signature as what we have expected. Importantly, when we look at the MCF10A with MSH2 knockout, it replicates the same signature as what we see in those patients that have DNA mismatch repair deficient. So we then also tested on the AL cell line, NUM6, also known to have DNA and MMR deficient, and we treat this cell line with thiopurine. It recaptured the same signature as we see in MCF10A and MSH2 deficient and the patient sample. So now we can establish a new signature that what we call thio-DMMR, really replicating, referring to those that have both DNA mismatch repair deficiency and also thiopurine treatment. So using this signature, we and also the previous thiopurine signature that we discovered, then we analyzed combined the cohort from the Shanghai cohort that we analyzed, as well as the target relapsed AL cohort. And we can see the thio-DMMR signature are present in both the US and Chinese cohort. And in most of the cases, they are all having the DNA mismatch repair, as well as relapsed during the treatment. So using the data from thiopurine as well as the TP53 mutation hotspot, we have once again to revise our relapsed model. So this is actually a data from one of the patients that we analyzed, and the signature mutational burden contributed to each signature was from the real data. So what we see here is that when the AL was first treated by induction, followed by maintenance therapy, the thiopurine treatment will lead to the acquisition of DNA mismatch deficiency mutations. Over here is a nonsense mutation in MSH2 in this particular tumor. And then because of the interaction between thiopurine and DNA mismatch repair, this will lead to the acquisition of P53 R240Q. So at the first relapse, this is still a subclone. But then when during the second relapse, when the patient was treated with the broad spectrum of the mutagens for treating the second relapse, the TP53 mutation clone was selected and expanded, leading to the second relapse. So we were very pleased to see that the the empirical data that we collected from this model actually matches very well with the mathematical hypothetical model created by Noel in 1976, depicting the evolution from the leukemia initiation to relapse during treatment. So the study was published recently, and again, the signature analysis and the connection between thiopurine DNMR to P53 R240Q mutation was done by Sam Brady, and all the experimental data related to P53 mutation was generated by Bingbing Zhou's lab in Shanghai Children's Medical Center. Now I want to switch gear, talk about our clinical oncology initiative. So this is an initiative that requires collaboration of multidisciplinary expertise from pathology, our computational biology, oncology, cancer predisposition, and subject experts in different diseases. So recognizing the importance of whole genome sequencing from our research studies, we have put a lot of emphasis to incorporate whole genome sequencing as part of the R3 platform, clinical sequencing platform. So we actually developed a pipeline in 2013 and 2015, performed a Genome for Kids feasibility analysis, and then 2017 is the real-time clinical genomics operation starts, and we also added 2 additional pipelines. One is called Rapid Turnaround Time RNA-seq, and this is used for supporting the Leukemia Total 17 clinical trial that established the eligibility of kinase inhibitor based on the presence of kinase fusions in leukemia samples. And the second is the 2 platform FFP-based exome and RNA-seq for mostly for brain and solid tumors. So our pipeline generate the whole genome, whole exome RNA-seq data from both tumor and normal sample, and then we collect a broad range of the somatic and germline variants, and then we perform automated variant classification, and then finally there's an analyst panel curation and panel review leading to the 5 category of pathogenicity, P, LP, VUS, likely benign and benign. And we will generate a tumor and germline-based report for each individual patients. So I want to mention a little bit about the pecan pie, which is a southern dish, but we use it to name one of our variant classification tools that were used for kind of triaging the variant from the entire, millions, hundreds or millions of variants at the start to a handful of them that could be used for analysts to do detailed curation. This was published and the method was published in Genome Research, and it was accessible from our cloud platform for free. So this is the analysis pipeline actually was used for our New England Journal of Medicine paper in 2015 on the diagnostic cases of pediatric cancer, and also the JCO publication of the pediatric cancer survivorship. And there's also studies that uses the tool to study family-based individuals, and this is one of the cases that we identified a loss of function mutation in ETV6 in a family study, and the standard analysis using linkage was not working in this particular case because one of the child does not have a complete penetrance of this phenotype. Now I want to mention about a little bit more on our Genome for Kids protocol. So this is to perform a whole genome exome and RNA-seq on both the somatic and germline tissues. And this time we have increased the coverage from the initial 30x to 40x for whole genome assay. As from our pilot study, we realized that in order to get at least 99% of the power for identifying variant with allele fraction greater than 10%, we need 45x coverage at the whole genome for tumor specimen. So I want to showcase two of the interesting cases that we analyzed, which changed the clinical care. The first is a child with a metastatic melanoma who had failed multiple therapies, including immunotherapy. So tumor was analyzed by our 3 platform sequencing, and we identified a novel MAP3K8 GNG2 gene fusion. So when this fusion was not reported before, but when we look at in the literature on the biochemistry assays performed on MAP3K8, and we recognize the deletion experiment shows that this will delete a autoinhibitory domain of MAP3K8. So we project this will cause MAP kinase activation independent of BRAF, and this melanoma actually do not have the BRAF V600E mutation. So we recommended MEK inhibitor, and indeed the patient had a total response, but unfortunately later developed with resistance. And we did sequence the resistant tumor, and turn out that the patient actually have gained an extra copy of this rearranged fusion. So it means that the resistance is developed by increase the quantity of the fusion gene. So next, we want to see how often we see this gene fusion in other melanoma. So this is in collaboration with Amita Baharami from Pathology Department, and who has a large collection of FP samples from split-stromal melanoma. So we performed the RNA-seq on this, and to our surprise, MAP3K8 actually come up with the top altered genes, either by gene fusion or truncation, that affect about 1/3 of the patients, and this fusion actually is mutually exclusive with all the other kinase mutations we detected in this cohort. And so the disruption or somatic event was either caused by gene fusion or truncation, and they all affect the loss of exon 9, which is again the last exon encoding the autoinhibitory domain. And from the RNA-seq, we can see a much reduced expression in exon 9 compared with exon 8. So use this profile, we're able to do a very quick screen on close to 500 RNA-seq from TCGA, and we recognized 8 samples have the same expression profile as what we see in pediatric cancer. And when we downloaded the full data set and performed the fusion gene detection, and we are seeing the same gene fusion or truncation as we see in pediatric cancer, suggesting that this event also affect about 2% of the adult melanoma. So currently we are collaborating with the chemical biology department to test new compounds targeting specifically on MAP3K8. And the study was published 2 years ago. The second case is about how to use somatic tumor genome whole genome signature for germline classification. So this is a very interesting case as the MUT-YH G396D mutation was considered as a fundamental mutation that affect mostly on the adult colorectal cancer. We discovered this mutation in a pediatric retinoblastoma sample, and importantly, we also see that there in the RBE tumor, there's a second hit, a loss of heterozygosity, leading to the loss of the wild type. So this is the tumor genome is actually homozygous with this mutation. And this retinoblastoma is unique in a way. It also has the highest mutation burden across the full genome for kids cohort. And also when we look at the mutational signature of this tumor, it has a very unique ROS signature that's absent in all the other retinoblastoma cases. So as MUT-YH is involved in repair ROH-induced mutagenesis, we believe in this particular instance, MUT-YH G396D actually is involved in the tumor development. So we are consider this variant as pathogenic in the particular setting. So our G4K study, again, was published in July, and I want to mention that the protocol was written by Dr. Kim Nichols, who's also responsible for the germline cancer susceptibility studies at St. Jude. And most of the analysis work was done by Scott Newman and David Wheeler in my department. So lastly, I want to give a brief introduction about St. Jude Cloud, which is a platform we created for allowing access, analysis, and visualization of genomics data generated from pediatric cancer survivors, and non-cancerous diseases. So we chose to use the cloud-based platform as people really appreciate that On the cloud, we only need to upload data and tool once, and every users can access the same computational infrastructure to perform the analysis. And we do know that computing cost on the cloud is still an ongoing challenge, and working hard on how to optimize our softwares to reduce the cost for our user. So the data contributing to St. Jude Cloud is coming from both retrospective studies like the PCGP, Pediatric Cancer Genome Project, as well as prospective clinical studies like Genome for Kids. And for the 3 major subgroups that of the cohort, we perform data harmonization, variant curation, metadata collection, and expression matrix. So the users can access the raw data as well as some of the intermediate data to perform the analysis. And these data were funneled into 3 apps. The first is the Genomics platform that we built with the DNA Nexus and the Microsoft Azure, so that the users can download data for free and perform analysis on the cloud using the infrastructure we provided. The second is Pediatric Cancer Knowledge Base, PCON, which we have collected published data from St. Jude and other institutions and harmonized them and curated them, put on this platform. And the last one is the visualization community, and we have put interactive visualization graphs initially created as static views for publication into this. And we are exploring functionality of really allowing research community to be able to do real-time analysis and sharing data and graphs with the collaborators through this platform. So right now, St. Jude Cloud has about 1.2 petabytes of the raw data from over 10,000 patients, and over 3,100 PIs have accessed the raw data from 100 institutions in 23 countries, and we have about 2,000 registered users. And there's also 10K unique users per month for accessing all 3 apps. Many of these are publicly accessible. So I want to say that this is a work, most of the St. Jude Cloud work was completed during the COVID, and we published our study early this year, and was supported by the St. Jude Cloud Blue Sky Initiative, and some of the tools we deployed was supported by my R1 grant from National Cancer Institute. I want to mention particularly one tool that we have deployed, which called SysX, and this tool was developed by Yu Liu, a former postdoc fellow in my lab, who currently is a PI at Shanghai Children's Medical Center. So this tool actually was inspired by the work from one PI at Dana-Farber, and I just want to show the 2 tails of non-coding regulatory variant analysis that this is the SysX tool was designed to support. So when you look at the large-scale pan-cancer studies, you can see that most of these papers, the result conclusions are actually quite disappointing. This is the Peacock study, that their main conclusions paucity of non-coding drivers in cancer. And the approach is that you need to do the significant recurrence analysis by checking with hotspot, windows, functional elements, recurrence of the breakpoints, or functional juxtaposition. So the yield was not very impressive. On the other hand, Tom Look from Dana-Farber 2015 published a very impressive study that really shows the oncogenic super enhancer formed by insertion in the non-coding region. And he only analyzed the one case, which is the Jurkat cell in TAL, and starting from the functional analysis, looking at the unique H3K27 peak that's present only in Jurkat. absent in other cell lines and suspected there's something going on, all he did is a PCR Sanger sequencing and discovered a novel insertion that can lead to the creation of MIP binding sites. And then screen for the patient samples, find this recurrent insertion, different insertion alleles, but all leading to the generation of MIP site. So I start to think, can I follow the footstep of Tom and looking at how we can systemize his approach, and can we discover non-coding variant in one sample? So what the approach we take is using allele-specific expression as a readout for candidate non-coding driver variant. So we know that the majority of the DNAs are diploid. So we have maternal paternal chromosomes and have a germline heterozygous variant that were indicating the dual allelic state of our DNA. And we have a somatic mutation here is responsible for cis activation of the gene. What we see is that in the RNA, we will see only one copy of the RNA instead of transcription of both copy of the DNA in our genome. So what we first have to do is really to create a model of balanced transcription, because this is the baseline that we will need to look at the deviation from this model in our analysis. So we chose to use a Gaussian plus binomial model as our null because we found that this actually captures the our theoretical sites that does not have ASE in the cell lines much better than using the other published approaches, and they appear to be quite robust even for regions with very low expression like 10x in RNA-seq. For this study, we also focused on the outlier high expression because we're looking at the the silencing event, which outlier low. This could be caused by alternative mechanisms such as imprinting, methylation, or nonsense mediate decay. So this is an overview of our pipeline, and you can see that it is a the utilizing single sample of interest as long as you have whole genome and RNA-seq, that we can derive allele-specific expression, and then by comparing with a reference cohort, we can derive the outlier high expression status, and combining these two signatures, we can identify candidate of 6 activated genes, and then connecting this with somatic variant that we'd be able to find the non-coding variants that are likely to be responsible for this. And this tool, in addition to accessibility in the St. Jude Cloud, can also get it from GitHub. So our benchmark analysis is on the 15 TAL samples. The reason we chose TAL is because, first of all, this is our first inspiration. And secondly, there has been a lot of functionally derived non-coding variants in T-lineage AL, as we've shown that at least 5 different publications have using functional-based approach to find these non-coding variants. So we can see whether we can replicate the findings published before using our algorithm. So indeed, our sensitivity is pretty high. We're able to find over 90% of the benchmark genes driven by non-coding variants. And we also discovered 4 additional novel non-coding variants in this cohort. So I just want to mention 2 of them. The first is a gene, PRLR, involved in JAK-STAT pathway. So we can see here is our whole genome. These variants are heterozygous, have both the reference and non-reference alleles. And in the RNA-seq, they become homozygous, monoallelic. And this is our index cases that's shown red here. It has the highest expression when we combine our 15 TALs with the target over 200 TAL data together. And we also find 2 additional cases of a target also have very high PRLR expression. So when we look at the all the genomic variants of these three data together, we recognize all three cases actually have focal deletions between IL-7R and PLRR. And when we superimpose the chia-PET data generated from Jurkat cell, we can see that these deletions actually will remove the boundary elements that separate the enhancers from IL-7R to PLLR. So this really shows that these deletion, intergenic deletion events actually are really responsible for activating PLR in these three TAL samples. The second case is a non-coding SNVs that we discovered in intron 1 of Tau1. And again, this is a recurrent event discovered in two cases, and we're seeing the same allelic imbalance and high expression of Tau1. And in this particular case, we were able to perform a luciferase assay and really showcase the activation, elevated expression in the mutant compared with the wild type. And we are also very fortunate that the PDX model was created for the patient sample. So we are able to perform ChIP-seq, H3K27 acetyl, and we also did the YY1 ChIP-seq because the mutation was projected to introduce a YY1 binding site. And indeed, we're able to see the two peaks centered around the mutant allele. And when we look at the sequence context of the ChIP-seq, we can see only the mutant allele, reads harbored mutant allele were aligned to this region, not the wild type, indicating mutation leading to the creation of YY1 binding and then the formation of a enhancer in intron 1. So the CISX was published last year, and since its publication, we're able to utilize this tool to make additional findings that are published, including a BCL11-1B enhancer in AML and mixed phenotype AL, MECON activation in AML, and also in our ClinGen, we found an interesting case that a MYC Notch1 enhancer known as NME, usually for activating c-MYC, actually has been hijacked to TAL3X3 through translocation and leading activation of TALX3. So finally, I will give a brief view about the visualization tools we developed. This is the Genome Paint, and we have the design of this tool has a very strong focus on capturing both the coding and non-coding variants in cancer. And the study was led by Xin Zhou, who again recently also become a faculty member at St. Jude. So the Genome Paint design is that not only can allow users to visualize a large cohort of the pediatric samples to find interesting patterns, but also you can go navigate to a lot of the details into individual samples. And in this particular case here, you can see that you can look at the RNA-seq coverage, recognizing a splice site variant leading to a loss of splicing in this particular RNA, and also perform some clinically related correlation either by single markers or multiple variants for looking at the potential clinical impact of these variants. So I just want to highlight one of the findings made as we are preparing the manuscript for Genome Paint. So as I mentioned, the NME enhancer was known to be a major driver in T lineage ALL, and was discovered by Adolfo Ferranda and several years ago. And a lot of the TALs have focal copy number changes, and indeed we observe here. So what intriguing when we put all the cohort of the leukemia together is that we see that NAM6, which is a B lineage AL, also contains this focal amplification of NME. And what we also find out when we link the DNA and RNA together, we can see NAM6 also is one of the highest MYC expression across the entire B lineage AL. So we have some high-C data generated previously from another investigation, and when we upload the data on Genome Paint, we're able to see that the MYC promoter actually interact with the NME enhancer in NAM6. And this is confirmed by designing Capture-C to confirm the interaction between NAM6, between MYC promoter and NME enhancer in this cell line. So another intriguing part is that we have seen a very broad RNA expression extended almost like a couple of hundred kb long in the cell. And usually we think enhanced RNAs as just a byproduct of RNA activity. But in this case, we see further deviation of ASE in enhancer RNA, even accounting for the copy number gains we have for this particular case. So luckily, we're able to reach out to Mark Valentine from Cytogenetics Lab, who has developed a unique technique called the serial RNA DNA fish that allow us to look at the MYC promoter as well as the enhancer RNA expression in the same cell. So what we found is that c-MYC is expressed only in one allele, and there's actually enhancer RNA cloud formed only in one allele, kind of indicating that this enhancer activity may have some function in maintaining the chromatin state that allow for rapid expression of MYC. So we are still investigating that. So this is my final slide. As you can see that I mentioned Tom's name a couple of times, and since our several publications between our two labs that St. Jude actually had encouraged us to consider to form a broad consortium looking at transcription deregulation across pediatric cancer. So I was very pleased to show that you can see some familiar faces with this audience, Kim Stegmeyer, Tom Look, along with Rick Young from MIT, Susie Baker from St. Jude, and myself. We formulated a 3D genome consortium for pediatric cancer cells, and so that we can bring our joint expertise in the genomics, in the gene regulation, and the model systems for brain, solid, and leukemia cells, and also dependency screening together, so we can understand how gene dysregulation contributing to tumor genesis, therapeutic, future therapy, and resistance in pediatric cancer. Thank you very much for your attention.
Hi, Jinghui, thank you for that terrific talk. There are some questions from the audience. Let me first start with Tom Luke's question. The progress of pediatric cancer genome and the disease gene identification over the last decade is phenomenal. Many years ago, Al Knudsen estimated that 25% of children with cancer harbor germline predisposition mutations. With all the results in the recent years, do you know roughly the percentage of children who develop cancer actually harbor predisposing mutations versus somatic mutations?
Yeah, Tom, thanks for this great question, and it's really great to see you here. I would say that currently, there's also autosomal dominant versus autosomal recessive mutations. If we combine we don't count for the mutational state of autosomal recessive. The number we have right now is about 12 to 15%. But if we only consider autosomal dominant, this is about 6 to 8%. I'm actually believe 6 to 8% is likely to be the real number, because when you look at compare the mutational frequency in pediatric cancer versus the control, the normal individuals, the recessive mutation rate is very comparable, but we see about 10-fold increase in the autosomal dominant cases. But again, you know, a lot of those have to do with looking at the secondary hit in the tumor. You know, the real, and like the case that I mentioned about MUT-YH, it's actually a recessive mutation, but in that particular context, it is involved in the tumor development.
Great. The next question is, how many of the newly identified driver genes in pediatric patient samples have targeted treatment available against them?
Yeah, I think the number is not very high, and it really varies from different diseases, right? So, and also the targeted treatment currently was very narrowly defined as kind of ways of, you know, like kinase activation was considered targetable. So like, for example, in leukemia, we believe the number is about 25%. And there's also, interestingly, some of the targetable variants actually could be in the non-coding region. So we have recently discovered FLT3, which is targetable. Some of the activation mechanisms found in BAL, especially in relapsed BAL, was caused by an intragenic deletion, again, removing these kind of the insulation, boundary elements, very similar to the PLLR. I think you know, people are looking into different targeting strategy, and I know Shirley has looked into the degradon, and there has been a lot of the work on looking at degrading these oncogenic gene fusions that mostly was leading to aberrant activation of transcription factors that's not deemed targetable, but that these new approaches can lead to you know, new targeting strategy.
Next question. Most of the non-coding variants by cis-X are from blood cancers. Can you comment on, I guess this is pediatric solid tumors, to see whether there are any recurrent enhancer variants?
Yeah, so I would say that you know, the pediatric solid tumor that we're focusing on, obviously neuroblastoma is one very prominent disease, as this is through collaboration with Tom. And we did, you know, at the very beginning, we used cis-X discovered TERT oncogene as was caused by rearrangement. And this is one of the cases that we were able to find by cis-X. There are a few oncogenes that we are currently under investigation. I just cannot reveal their name now. And one collaboration is actually also involving, was brought in by Kim Stegmeyer, connecting us to a group in Dana-Farber, who's actively working on an oncogene we believe works, play a central role in both brain tumor and the solid tumor. I just cannot reveal their identity at present.
So I have a related question. If these cis mutations are mostly in the blood tumors, what normal sample do you use? Like, because you will see them both in the whole genome sequencing and the RNA-seq. And so how do you know this is a germline versus somatic?
Yeah, this is the germline versus somatic is relatively easy because we always sequence the germline samples. So we can figure out, use it, you know, if it's not in the germline, but only in the tumor, then it becomes, we can know this is somatic. But the germline, you know, if the variant is germline and associated with cis-X, it does not really diminish its role. So I just want to say that, you know, for example, the tau1 intron mutation, we actually found one case, not the 15 TALs, but in one of the children's oncology TAL cases, it's actually a germline mutation that we observe in this. So their frequency is much lower than somatic mutation. But we do see there are germline events playing similar roles as the somatic variants.
For a blood cancer, what tissue do you use to provide that germline reference?
Oh, you mean the normal, right? Yeah. So we don't have, this is where like the public data resource is lacking, especially for pediatric tissue, because adult cancer you can use GText. So for us, we have to use GText as a control, but on the other hand, I heard that NIH may be funding to a developmental GText, so that could also increase the kind of developmental stage match the normal tissue for us. So the other kind of the control we use here is within the tumor cohort. So the reason this is that we were looking for aberrant activation. So it is not all the tumor samples will have this event. Only a subset of the tumor samples will have this event. So you can look across the tumor cohort to separate those that have the mutation versus those that do not have the mutation. Those that have the aberrant activation versus those that do not have the activation to detect the status. And again, this is, these are the association, not definitive. So this is why the experimental evidence is always crucial, right? That's why finding the non-coding variant is so much harder than the coding variants, because you have to prove causality and perform the experiment.
Yeah. I noticed that you have a GR, nuclear receptor NR3C1C2 mutation in the later relapse samples. Were these patients treated with dexamethasone before they developed the relapse? And if not, could we treat these patients with GR mutations with dex to overcome that resistance?
Yeah, so I think the GR mutations will be resistant to glucocorticoids, right? So we would recommend avoiding this drug. And there's also some important. This is actually you raise a real great point. Kevin Shannon, when we submit the blood paper, Kevin Shannon from University of UC San Francisco, he built up a mouse model and on TAL and treating these mouse with glucocorticoids, and they developed the same type of mutation as the NR3C1 as we found in pediatric cancer. And also, I didn't mention too much, but the NR3C1, at least these, we did see a couple of the mutations in the non-coding region appear to be related to the loss of, reduced expression. As CysX in our current version is only looking for oncogene, we haven't looked deep into the silencing effect. And we can take a closer look to see there's, truly any events that due to the loss of the promoter and other regions that are leading to the reduced expression during treatment.
Let's see, I think there's another, let's see. Do you think THEO treatment plus MMR deficiency can generate hypermutations that are amenable to immunotherapy?
Yes, indeed. This is a very acute observation. I think you can see that in my slide about the last one when combined the US and the China cohort together. Those that with the highest bar, the highest mutations, are those with the THIO and MMR deficient. So they definitely will be, have more abundance. And then the mutation numbers you can see is some of these, is very high compared with other pediatric cancer. So I definitely anticipate there are going to be more abundance of neoepitopes in these relapsed cases, and whether it's the checkpoint inhibitor or a CAR T or other immunotherapy approaches. This is where we hope that the ideal situation is we're able to predict which samples can lead to these kind of cell DMMR mutation prone to these, so that they're not going to be exposed to really toxic chemo in the first place. Then it can go directly on immunotherapy to avoid all the toxicity that could be caused on these patients. So that's kind of our hope in the future, our dream in the future to accomplish.
Great. Yeah, I think our time is up. So I would like to thank Dr. Jinghui Zhang again for this really wonderful seminar, and also for the really nice St. Jude Cloud, that data resource and visualization tools, at which I'm sure we will all benefit from our respective research. So thank you again.
Thank you, Shirley. Bye, everyone.
Yeah, everybody.